Hello guys, so welcome back in another video tutorial for microbiology with Shagnik. So in this video tutorial we will going to talk about the first type of processing of antigen by MHC molecules, because in previous video I have told you various times that antigen will be processed by the MHC molecule, and a short part of the antigen that will be presented to T cell, right? So in case of MHC class 1 molecule, it was 8 to 10 amino acid long peptide, and in case of class 2 molecule, it was 13 to 15 amino acid. So today we will going to see how MHC class 1 molecule converts a big protein into a small 8 to 10 amino acid containing peptide. Okay, and the process how MHC converts a big protein into small peptide of 8 to 10 amino acid is known as cytosolic or endogenous pathway. Okay, and now let me, before I going to talk about this, please tell me that what kind of antigen MHC class 1 molecule deal with. So I hope you remember that I have discussed with you that MHC molecules are just like our fathers, right? So they have to handle their house problems first. So MHC class 1 molecule will going to deal with endogenous antigen, right? And MHC class 2 molecule will going to deal with exogenous antigen, right? So today's antigen processing is all about the processing of endogenous antigen. Okay, so the cells that express MHC class 1 molecule, I have told you, are known as target cell, and most of the cases these cells are nucleated cell, right? Clear? And this nucleated cell will going to interact with CD8 surface molecule containing T cell, right? And CD8 surface molecule containing T cells are also known as cytotoxic T cell. Clear? So they will interact via the T cell receptor. Remember, it is known as T cell receptor. And let me clear another more thing, that when I will discuss with you about the processing of antigen in case of MHC class 2 molecule, there you will also find the term TCR. Okay, both are the same. Okay, and I will going to also talk about the details of T cell. There I will also mention that concept of TCR, so don't worry. But for now, please remember that the TCR in case of cytotoxic T cell and the TCR for helper T cell are same thing. Both will going to interact with MHC molecules. Okay, and if I zoom this picture, so this one is your antigen. Okay, but now let me introduce another important thing. That does not mean that all the antigen will be processed in this way and presented in this way. I have discussed about in earlier times when I have taken the lecture of medical microbiology. I have talked about the concept of super antigen, right? There they will not be presented to antigen after being processed by MHC molecules, right? There they will directly presented, okay, in that way. Okay, that is why I want to just mention that please don't consider that all kind of antigen will going to processed in this way. It is not, but most of the cases it will be processed by MHC molecule and then presented to T cell, right? If it is MHC class 1 molecule, then it will going to interact with CD8 cell surface containing T cell, and if it is MHC class 2 molecule, then it will going to interact with CD4 cell surface containing T cell, which was helper T cell. Okay, so now please take a screenshot of the thing that I have mentioned here. That will help you to make a note of it. So I hope you have taken it. So now another important thing I want to mention, that is the processing of MHC class 1, processing of antigen by MHC class 1 molecule can be prevented by any kind of protein synthesis inhibitor. Okay, because remember, here I have mentioned that class 1 presentation required internal synthesis of viral protein. Okay, that means the target cell must be infected with live virus, and class 1 presentation on the cell surface can be impaired or can be hampered when protein synthesis is blocked by inhibitor sometime. Okay, that is quite important point, that the target cell for expressing MHC class 1 molecule, the target cell, that means this one, must be infected with live virus. Okay, and that is why they are dealing with endogenous antigen, because most of the endogenous antigens are viral antigens or cancerous cells, right? So that is the concept. So now please take a screenshot of this, then we will move to the next one. I am again want to mention that please take a screenshot of this, because I will going to explain the entire thing with the help of a diagram. So please take a screenshot. Okay, so now this picture will going to help you to understand about the concept of endogenous processing or cytosolic processing, or how antigen are processed before it is presented with MHC class 1 molecule to CD8 cell surface molecule containing T cell known as our TC cell. Okay, so let's start. So first of all, there are two different things. One things are going on in cytosol and another things are going on in rough endoplasmic reticulum lumen. Okay, so let's see. First we're going to talk about how class 1 molecules are formed. Okay, so first of all, remember the arrangement of class 1 MHC molecule, that means the arrangement of alpha and beta domains. So alpha 1 will be there, alpha 2, alpha 3, and there was our beta 2 microglobulin, right? That was the arrangement. Alpha 1 and alpha 2 domain interact to form the peptide binding group, and beta 2 domain interact with alpha 3 domain via non-covalent interaction, right? So now we will going to see how they are formed. Okay, that means how they are arranged. So first of all, MHC class 1 alpha chains are always together. Remember this. That means alpha 1, alpha 2, alpha 3 are formed together. And if you see the picture that I have drawn in case of MHC class 1 molecule video, there I have described the entire thing in this way, right? And beta chain was situated separately, right? So in this way you can understand that the entire alpha chain was together from the very beginning. Okay, later it was somehow beta 2 microglobulin attached. Okay, so today we will going to see how beta 2 microglobulin attached with MHC class 1 alpha chain. Okay, so let's see. So there are various chaperones involved in this process. First chaperone is calnexin. Okay, so MHC class 1 alpha chain and calnexin will going to exist together. Okay, that means there will be a complex between MHC class 1 alpha chain and calnexin. Okay, not only these two, along with that there will be another molecule named ERP57 that will going to make a complex. That means first complex was between ERP57, MHC class 1 alpha chain and calnexin. Okay, now remember this ERP57 has enzymatic activity. Okay, and this ERP57 calnexin together promotes proper folding of MHC class 1 alpha chain. Okay, and remember very important thing, that for protein it is very important to be properly folded, right? So it is important that ERP57 and calnexin together assembles with MHC class 1 alpha chain. Okay, although they will going to remove in the next step, that is the different case. And remember the entire thing will be going on in area lumen. There will be nothing going on in cytosol. Okay, the entire thing that is the formation of MHC class 1 molecule will be going on area lumen. Okay, and now what is the function going on in cytosol I am going to talk about. So after that, beta 2 microglobulin will going to enter. Okay, this small black color protein, as you can see. Okay, so now what beta 2 microglobulin will say that I will not going to exist with calnexin and ERP57. They are my enemies. I will not tolerate them. Okay, under such circumstances, what MHC class 1 molecule will do? It will ask calnexin and ERP57 to please remove, right? So beta 2 microglobulin facilitates the removal of calnexin and ERP57. Okay, it is just like girls simply cannot tolerate that their boyfriend told will gossip with someone else, right? So I am joking about that matter. That's I am talking about those girls. Those are too much possessive towards their boyfriend. So beta 2 microglobulin is the girl who is too much possessive towards a basic class 1 alpha chain. Okay, and that is why it cannot tolerate the presence of calnexin and ERP57 in presence of MHC class 1 alpha chain. That is why they beta 2 microglobulin facilitates the removal of calnexin and ERP57. So I am just joking. I am joking so that you can remember it, right? So quite funny, right? So now after that, beta 2 microglobulin will also allow the entry of tapacin and calreticulin. Okay, so calreticulin and tapacin are another two chaperones. That means although girls do not allow her boyfriend to meet with other girls, but the boyfriend have to tolerate his girlfriend's friends despite he don't liked it. There I am just joking about that. So beta 2 microglobulin will facilitate, I am just try to explain it in such a way that you will not forget. So beta 2 microglobulin will facilitate the entry of tapacin and calreticulin. Okay, on the other hand, beta 2 microglobulin also facilitate the removal of calnexin and ERC57. That is the thing. Okay, and sorry for all the joking. So now the entire combination was ready. Okay, so this is the MHC class 1 molecule. That means MHC class 1 alpha chain. This one is your tapacin. This one is calreticulin. This one is beta 2 microglobulin, and this one is your ERP57. Okay, so now we will going to talk about how peptides or how antigens are processed into 8 to 10 amino acid containing peptides. Okay, so now focus. So first of all, what I have told you, that the antigens that will going to presented by MHC class 1 molecule to TC cell are endogenous antigen, right? So endogenous antigens are in intracellular sources. So remember, the first step is the complex formation, and this complex is between ubiquitin and endogenous antigen. Ubiquitin is a protein. When it form a complex with endogenous antigen, just like that, it will facilitate its degradation by proteasome. Okay, this proteasome is a thing that is present inside cytosol that can facilitate proteasomal degradation, okay, via ATP dependent process. Okay, so what I have told you, that first endogenous antigen will going to form a complex with ubiquitin. Okay, now this ubiquitin endogenous antigen complex will going to enter in proteasome. Let me write it, proteasome. Okay, now this proteasome will do proteolytic degradation. That means this proteasome will going to break the peptide bonds that is present in endogenous antigen. Okay, and it is a ATP dependent process, remember it. Okay, and as a result the peptide becomes near about 8 to 10 amino acid long. Okay, so now remember one thing, in most of the cases proteasome tried to convert the endogenous antigen into 8 to 10 amino acid containing peptide, but in some cases they produce peptides of little bit 13 to 15 amino acid or 20 amino acid long. Okay, but I have told you that MHC class 1 molecule will going to present 8 to 10 amino acid because the peptide binding group formed by alpha 1 and alpha 2 domain only have the ability to interact or only have the ability to allow the binding of 8 to 10 amino acid long. Okay, then how a peptide molecule having 20 amino acids can be fit with MHC class 1 molecule? This is a very big question, right? Under such circumstances, there is a protein present on area lumen that will going to play an important role. But before that, the first thing is the peptide that was produced by proteasome have to be transported to area lumen, right? Because MHC class 1 molecules, now I can say them as MHC class 1 molecules, are present in area lumen, and the peptide that was processed by proteasomes are in cytosol, so it have to be transferred. Okay, now remember this transfer process is mediated by tap transporter. Okay, this tap transporter, remember, is a ATP binding protein that is pre-optimized to transport peptide containing 8 to 10 amino acid. Okay, so it will be transferred via tap. Okay, now sometimes tap also transports peptide having 13 to 14, 11 to 12 amino acid containing peptide. Okay, but I have told you that the peptide binding group formed by alpha 1 and alpha 2 domain only allow peptide having 8 to 10 amino acid, right? So there should be something that will process the 11 to 13 amino acid or 13 to 15 amino acid into 8 to 10 amino acid containing peptide, right? So this protein here is ERAP, that is endoplasmic reticulum aminopeptidase. Okay, this protein will going to convert all the peptide into 8 to 10 amino acid containing peptide. Okay, now it will be presented by MHC class 1 molecule to CD8 T cell. Okay, and before that process all the other proteins, for example ERP57, your tapacin, your calreticulin, all will be removed. Okay, only beta microglobulin and MHC class 1 alpha chain will remain. Okay, now another important thing, remember here the protein tapacin will going to play important role. How? Because by its name it is easy to guess that tapacin means tap associated protein. Okay, so this tap associated protein will bring tap transporter to the close proximity of class 1 alpha. Okay, so that class 1 alpha chain can easily take the antigen from the cytosol to its peptide binding group. Clear? So that is the entire process of endogenous processing or cytosolic processing of antigens that is going on in case of MHC class 1 molecule. So for a quick recap, first of all, MHC class 1 alpha chain form a complex with calnexin and ERP57 that is necessary for its folding, remember. After that, beta 2 microglobulin will come into the picture, but it cannot tolerate the friendship between MHC class 1 alpha chain and calnexin, and as a result beta 2 microglobulin forces calnexin to remove. Okay, but beta 2 microglobulin in turn also facilitates the entry of tapesin and calreticulin. Okay, and now the entire complex is formed. On the other hand, another thing is going on inside in the cytosol, that is, if complex called endogenous antigen ubiquitin complex is formed, this complex will be processed by proteasome, which is a proteasomal degrader that can degrade protein via ATP binding process or ATP mediated process. Now the peptides that are produced are transported by TAP transporter. Okay, and now all the peptide which is quite excess in their size are further processed by endoplasmic reticulum aminopeptidase that will produce a peptide of 8 to 10 amino acid. Okay, now it will be presented by class 1 MHC molecule to TC cell, right? So that is the entire idea of antigen processing by MHC class 1 molecule. And remember, this processing is known as cytosolic processing or endogenous pathway, endogenous processing of antigen, and it is quite clear because here involvement of cytosol is observed, right? So I hope this video is now very clear for you, and it will be also helpful for you. And if it is helpful, you can like it, share with your friends, and subscribe to my channel to get more videos like that. So thank you for listening to this class.